Welcome back. So, in the previous video, we show you how to perform a statistical analysis using Lima and how to visualize the results. But now, how I can analyze this further? What information I want to understand those expressions, those abundance values that the analysis gave me. What other insight can I have that can help me to understand that those changes are really biologically relevant? So a way to do that, there are of course several tools to do this task, but one tool is Ingenuity Pathway Analysis, or IPA, and it's the one that we are going to present to you in this uh, hands-on demonstration on how to use IPA. So something very important is after we finish our analysis in R, we can export the results. We can export the table as, we can export it in many formats, but since the CSV is the one that we gave, we can export it as a CSV as well. So for this one, we're going to say use the function write.csv. So this function stands for I want whatever output I have, whatever output I want to export, to be exported as a CSV file. In this example, we are going to export the results from the top table that we ran before visualizing the data, the statistical results for the total of 1884 proteins. And so that's why this is results variable in here. We are going to say that the output CSV is called demo underscore results.csv. And since my row names are the protein exceptions, now I want them to be as a column. So we set this as true. If we set this false, the row names will not be exported in the CSV file. So once we have the code ready, we execute it, we click run, and it shows that run successfully. We go to our folder where the results are located, and these are the results file. We're opening it in Excel. We're going to tweak this file a little bit because IPA works very well with tables as well, but we need to add something else. Before doing IPA, we are going to just give this column again the accession column name, and with that we save it in case that we cannot undo anything we did already. And for the IPA analysis, we are going to use the accession, the fold change, and we can provide p-values as well. We can give the two of them. We can give p-value and adjusted p-value as well. So all of the other columns we can delete it, or we can filter them out once we upload this dataset to IPA. So first, instead of saving it as a comma separated value, we are just going to save a copy as a spreadsheet of Excel. And we are ready to submit this data to IPA. So I already have IPA up and running. So this is the main interface of IPA. As you see, it has a very graphical user interface. This main part over here is the search engine. Here you can search for genes, or chemicals. But these words are just that, just words, because you can give as well protein names, protein accession, and many other identifiers of a gene and or protein, as well as chemicals. Diseases and functions related to diseases and diseases that are related with functions. They are very interconnected between them. So functions are mostly based on the terminology of gene ontology. Diseases are, as the word explains, diseases, mostly cancer in overall, but you can have different annotations of other diseases. For example, um, let's say dengue fever, uh, metabolical diseases as well, inflammatory responses in terms of functions. And in here, in this tab, you have pathways or toxicity list. This one is one of the main features of IPA because it provides a very well documented pathway representation of they have a good repository of the canonical pathways, quote unquote, in terms of, let's say that you want to study a pathway related to cancer. If you want to study uh, protein kinase, for example, these kind of analyses are very good for that kind of approach. So you want to pay attention more with pathways. So in here, you can search if you know the pathway. You can type the pathway. The same with here, diseases and other functions, and in the same in here. But first, you need to click the tab that you want to search, and then type the name. So for example, before we upload the data, let's say that I want to search for a particular gene. I have EGFR as my gene, and I click search. So after I click search, it can take a while. It can take a couple of seconds to search, and I give this kind of result. If we expand it a little bit, we can see that it has the symbol that I given, and that's why it's highlighted in yellow. And then match terms that I can give that work good, compatible with EGFR as well. Can be EGFR in lowercase, EGFR1, and so on and so on. Those are the ambiguous names that this gene has been given in the literature. Synonyms as well, the description of the name, where it's mainly located, what type of protein or gene is this, interactions. If we click that, it will show interactions between EGFR and other nomenclature, for example, other genes, other proteins, other functions, diseases, and or pathways. So let's try a moment to do that. It can take a little bit of while because if it is, as of course, it has too many. So this will be a very small and very short sample of creating your own networks without data. So this will be a very quick demonstration of how to do it without data. So you have EGFR represented as a node, and let's say that, let me just enhance this. In here, in this part, you want to make a network, not a pathway. This is a very important. Uh, if you have a pathway, you can represent it as a network, but not the other way around. Pathways usually gives directionality. Pathways give usually step-by-step process that are required. So, for example, if you have EGFR interacting with protein B, that's the first reaction. So, protein B then reacts with protein C up to protein C, for example. It represents directionality it represents order, not the other way as the networks represents the data. But you may represent a pathway as a network, but it will not give you directionality. What starts, what it ends, uh, what is the secondary interactions, what are the products, what are. So it doesn't give you enough information so we can construct a orderly cascade or orderly process. So the pathways serve mainly for that purpose in terms of how IPA chose the data. But networks are essentially very good to represent what genes are associated with a disease, what genes are associated with a function. So in this case, we can do it both ways. We can say first that EGFR is alone here, is just this single node with no interactions. So now we are going to say, well, I would like to have a maximum of 10 genes or molecules at a time. So this you can modify it as a big number or as lower number as you want. The bigger the number, the more complex, the more uh, diverse and less understandable becomes. It doesn't matter that it doesn't have good information. It's just it gets too convoluted, too much information to analyze at once. Usually, you will end up with small number of genes and proteins to work with, and that's why it's very important to actually study the analysis as we did in these sessions, to first do the statistical analysis, filter down from in the example that we provided for Lima, 1884 proteins to at least 3, 4, or 10. Usually, you will end up with less amount of proteins or genes than the ones that you gave as input. But those are the ones that are saying that this data is relevant. This is the proteins that you will need to pay attention to. So a very quick demonstration on how to create this is to say, well, I just want 10, and we can specify as many things as we want, but since this is a very quick demonstration on how to do it without using your dataset, we're going to say only for humans. This is another critical step. Since IPA works very well with humans, rat, and mouse, it doesn't do uh, too complex with other organisms. So when it says uncategorized, it just says anything else. Literally says, well, we're going to find orthologous proteins that are similar but in other organisms. Or let's say that you have other organisms and you want to say, well, I have this list of significant proteins that are from yeast, from a parasite, let's say Plasmodium berghei. And you have a list of proteins that are specifically for that particular organism, but you want to see if that list of proteins are orthologous in human, mouse, or rat. So that's a way that you can do it. You can try to enrich your proteins using IPA for that matter. But it's very well done for human, mouse, and rat. So in this case, we're going to select human. We can select mouse and rat. Tissues and cell lines. This, as if you want to be as specific as you want, it will take longer the time to run. It will just take a long time. But in this case, you have, for example, most of this data, and this is important as well. Most of this data is from literature base. The IPA do most of the enrichment analysis by having a group of curators that are reading literature and enriching the interactions, not entirely manually, but they are updating this continuously. And as well, IPA has as well this text mining algorithm that literally takes into the context of the literature the interactions between protein to proteins or gene to genes, genes to diseases, and it takes the action verbs that are in the literature and tries to make the connections between them. So that's a way that IPA works around how the IPA creates networks, how IPA construct their knowledge base. As you can see, it has a very extensive, long date of a availability range. So you can see that from the last century, at least from 1954 to this year, we can say since December is not arrived, from January of 1954 to January of 2020. And that will be the range of all the publications that have been published related to the gene EGFR and the other proteins that we have been working, that we want to see what they are. And as well, for diseases, we are, let's say, interested in cancer. And that can take a while. Let's see. So we at least are expecting having 10 genes or chemicals associated with EGFR. And this here helps to change the layout. So usually, and one of the most preferable ones is the organic. We just disperse the nodes as organized as it can be. So a little quick introduction is solid lines represent direct interactors with EGFR. Dashed lines like this represents secondary interactions. It will be indirect interactions. You need something in between these two genes to happen so it can interact to EGFR. So something that we can say is we can double click this annotation in here, and all these nodes, as it shows, are data points, are proteins or genes that are connected between them. And the lines are called the edges. So if we click all of them, let's say that if we can, all of these are clickable. Let's say that I can click this again. If I click this, inside that edge is this annotation. So I have the same type of data for each edge of this network. And this is the annotation and the type of annotation it represents. Maybe activation, be upregulation, be causation, whether that be. If we want to see the reference of that interaction, we can click View Relationships between AGK and EGFR. When we click, we are redirected with an HTML report of the actual literature that is being cited that says this interaction. If we go to IPA as well, again, and we click this one, this is a new and a different type of edge, a different type of line. This line, it represents protein-protein interactions. When we don't see an arrow like this, when it's a very solid line, usually it represents protein-protein interactions. And when we double-click this, it will confirm that it's a protein-protein interaction. This is the interaction, the annotation, and we can as well access this. So every network that you create is two things: is unique and At the same time, it's well annotated. It needs to have literature or even prediction, some sort of validated information that confirms this interaction is happening causally, really. So with this demonstration now, we can start our own analysis with the results that we obtained from the Lima statistical analysis. So in here, after this little introduction, in here we have our project manager. In here you will have the list of all the projects you are working on, all the bioinformatic analysis. It doesn't matter if it's proteomics, microarrays, RNA-seq, microbiome, uh, mutation analysis, in a degree can provide information. So in this case is proteomics. So in here now I would like to show how to create a folder, how to upload data, and then what kind of analysis we can perform with the data that we are uploading. So a way to upload data sets are two ways: in new and upload dataset. But first, we need to create a folder for this analysis. So a way to create it is by clicking new and project. The other option will be inside my projects. We can say new, new project. And now we can give name. This case will be demo analysis Lima results. In here you can type note if you have microarray or RNA-seq data, or you want to say, oh well, this data is TNT10-plex. That's something that is helpful as well. If you have microarrays, you can type in here, for example, Affymetrix SHIP 1.0 of human or something. So it depends on the data that you have. So in this case, demo analysis Lima results from TNT-plex, and then we can create that folder. It can take a while to run, and it has already created automatically for you. It will create all these folders. It creates dataset files, analysis, comparison analysis, so on, so on. So the top 5 most important sections of this directory is dataset files. Is where you are going to see the tables that you are uploading or you uploaded already. The analysis corresponds to the folder where RU analysis will be stored. That will be discussed later on. Comparison analysis is good if you have one or more analysis and you want to compare the expression pattern of the list of genes that you uploaded, and you want to see, well, let's say that in my comparison A, the gene EGFR is upregulated, but in my comparison B, EGFR is downregulated. So you can see that pattern and how those expression values, those abundance values in terms of protein analysis, are significant to predict changes, causal changes in pathways. My pathways is very important if you have a list of proteins or a list of genes that are analyzed in a canonical pathway, and we are going to discuss that later on. My list will represent literally what it says: list of proteins, list of genes, list of chemicals, list of any omics type of list that you want to analyze using IPA. And that's something that is helpful when you have already a data set and you want to see specifically a protein that you are not sure how it's interacting in your data set, but you don't see it in your data set, but you want to predict what proteins from your data set are interacting against that protein of interest or that list of proteins or genes of your interest. So essentially now, the next thing we're going to do, let me just clean the search, is to now upload data in here, in this directory. So again, two ways to do it is dataset files, upload dataset, or in here, upload dataset. The convenience is if you do it in here, it will be directly in the directory that you are selecting. If you're doing it in here, you need to specifically give which ones of this list is the one that you want to submit the upload. So it's better to do upload dataset and then go to the directory where you save your file, and we are already in the directory, so we're just double clicking it. Now it will redirect to this dataset upload section. So it already recognized that it's in flexible format, but the main importance is that the flexible format allows you to edit the columns you want to analyze. So that's why flexible format is the default. Actually, this is actually default. Most of the tables we provide to IPA, they already have headers. That's why we took a little bit of time to edit this column with the protein accessions. We just wrote the accession name into the column name. There. So in here, since this is not an array, we don't say that it's not applicable or not specified. And now in here we have the up drop down menus that represents the columns of our dataset that are the results of our analysis from Lima. So this one will be our ID, and it's actually already running. It detected 846, but we can say if they match it correctly. Since this data is from UniProt accessions, it makes sense to just leave checked UniProt SwissProt accessions. And now every change that I make, it will just reprocess the whole list. It will update the list that I provide. So it's still the same because all of the accessions are from UniProt, not from GenPet. In this, we are going to say this is from observation 1, and this is, of course, log ratio. Which is the same as log fold change. And here, the same will be observation 1, already detected that is the raw p-value. And here, in this one, we need to modify a little bit because this one is actually the FDR. And you notice something in here. You are allowed up to a maximum of 6 different observations. What that means? Well, now we already selected that all this data that we are highlighting is from one observation, is from one case and control scenario. It's just from one experiment. But if we have up to 6 experiments, we can upload the whole thing with 6 different fold changes, 6 different p-values, and 6 different fault discovery rates. After we have everything set and we are satisfied with the settings and parameters, we just click Saved. It's going to give us a message. Usually this is not critical, this is not it's not important. It will be relevant if you have metadata to provide. But in this case, we just click OK, and it will not be a big problem. It will not be a problem at all. And then it gives us the opportunity to say, well, do you want to save it as the number I was uploaded, or you want to give other name. So in this case, we can leave the name as we are going to upload it, and automatically is saving it in the directory we want. If for some reason you didn't create the directory previously, you can save it in here, and it will create it for you and it will save it for you in that directory. So now it's uploading the table, it's uploading the whole results. This can take a little bit of while. Now it's annotating and enriching those IDs that we have provided into names. So we notice something in here. We notice that the parameters of fold changes needs to be recalibrated, but that's something we can modify, because if we, in this case, we are satisfied with everything we are seeing, we are going to do something so these values are modified. We need to rescale them. So a way to do that, later on we are going to do that, but for the moment we have all the information and even more that IPA provides. So for each accession, it detects the name, the entree's gene name, where it's located, the type of protein, and if it has information about drugs that interacts against that protein, it will give it to you as well. So this in here, it says that the data has 9 different tables, and you can see the ones that are mapped or identified with IPA, the ones that are unmapped, and we create an other tab and the total of all of them. Now, after this step, usually the first analysis we are going to perform is the core analysis. To do that, we just go to Analyze, Filter Dataset, and we click Core. So core stands for literally what it means: the essential enrichments for all this list of proteins or genes or chemicals that you wanna enrich. So in here, after we select the core analysis, it is asking, based on this data set, which core analysis type would you like to run? So the default will be expression analysis. If you have toxicity analysis or even metabolomic analysis, can be analyzed using IPA. So in this case, it would be expression. And we would like to use the log ratio to base the measurement of expression. After that, we click next, and now it will give us a bigger prompt of multiple options and additional settings that we can preset before we run the core analysis. So in here, we would like to run interaction networks and causal networks. So we leave it as checked. We would like to study everything that these proteins are interacting with. Usually, another terminology that the community uses is fishing. We are fishing the information as much as we can get of the list of proteins that we want to enrich. So most of the time we leave this as default because we are just exploring the proteins, the list of proteins we are analyzing. In here, most of the time we select experimentally and highly predicted. So experimentally observed is usually the ideal annotations we would like to have. But sometimes highly predicted are just automated annotations that need to be verified by a curator, but are enough to get a glimpse of what that protein might be interacting with or what information we are or we would like to see. But usually experimentally observed is the ideal because it's the one that has been published manually and automated curated, but mostly human curated as well. We would like to say, well, for this case, since we don't know what organism and that data, this data is since it's a demonstration data, we are going to explore all the organisms that IPA has available. So we just leave it all checked, as well as tissues and cell lines. And this is just an additional layer. We can deselect or just leave it as it is, the mutation analysis. For demonstration purposes, we will leave it as default. And in here is where I was mentioned previously that we can modify the cutoff values of expression. So usually you can filter this out before uploading to IPA, but IPA assumes that you need to filter out the data, and it's something that is additional step that you can perform in here. You can arbitrarily do this, but since Lima we have been working with a 1.5 fold change or a log to fold change of 0.58, we're going to say, well, give me everything between negative 0.58 up to 58. Anything below or equal negative 0.58, or everything over or equal to 0.58. And we would like to say, well, give me only the 0.05 proteins with this p-value, and as well the ones that are 0.05 of this p-value. We can just recalculate to see how many of them they are. Of course, we are very conservative about this, so we can just remove this threshold and recalculate it with only using the FDR. We end up having only 2. That means that the other one from the 3 proteins that we had previously in Lima, one of them wasn't identified in IPA. We have only 2 proteins that are that were identified in IPA. Something as well that we can do is to be more flexible with this cutoff, and instead of using the FDR to have more information but still considering some sort of significance, we're going to be less conservative and say, well, give me everything that is in this range of fold change but with this p-value. And now we end up having 3. So it's, of course, from 1884, we end up having a list of only 3 proteins considered significant with a fold change, with a log to fold change of 0.58 and a p-value of 0.05. So this data is already for analysis. And in here we can see what proteins are significant. And if we are from a biochemistry background, from a more biological background, we can say from the information we have already that these 3 proteins are related in some kind with electron transport change, Krebs cycle interactions, but since we have IPA to confirm that for us, we can check, for example, if those proteins are already in the functions we are predicting, or in what other functions these proteins are interacting with. So After we have everything set, we just click Run Analysis. And this, we can modify it. Usually, my recommendation is when you use IPA to first add what type of analysis you're doing, dash, underscore, space, it doesn't matter. Uh, but usually we use it with a dash, call results, the date, and time. But if you want to be very, very specific, in this case, we leave it as it is. Enough information to know what we did. And then we just click OK, and it will be running. So this will be the first part of this analysis. In the next video, we are going to interpret the results of this analysis. So thank you for listening. Thank you for watching. We'll see you in the next